All right, Anna here, and we're going to talk about part 4 of the nervous system on neurophysiology. We're going to talk about the different type of gates that you have and how they interact with the plasma membrane. All right, Anna here. I've already said that. Why am I saying that again? So before we get going too far, what I want to talk about is the plasma membrane and kind of refresh your memory on that. So here you've got your little phospholipids. All right, that's forming that double layer of the plasma membrane right there. Okay, now the next thing I want you to notice are the different types of gates, or proteins, not gates, the different types of proteins. These are transmembrane proteins, so that are embedded in the plasma membrane. They have different functions, and this is review. And you do need to know these, okay? So like number 1, this particular transmembrane protein is going to respond to a chemical messenger that's going to change the shape of this thing and then trigger things to happen inside the cell. Chemical messenger here, when it clicks onto this is an enzyme embedded that triggers it to break down. So these are transmembrane proteins that are interacting with cytokines. Nothing comes into the cell, so there's no column or tube in between it, but it does trigger things inside the cell or it breaks things down. Then over here, let's look at number 4. So we talked about antigens before. We'll talk about antigens again in AMP2, especially with your ABO blood type system. So this would be like a protein marker, okay? So a protein marker that sticks into the plasma membrane, and it basically identifies you as self, okay? And then here's another interesting one right here. This is one that allows things to hook onto it. So this is another cell over here. These two ends match up and they can adhese, so an adhesion molecule. You'll talk more about this in pathophysiology or in microbiology, so that's an important cell as well. Now, these are the next two things right here. This is a passive gate, okay? Passive, okay? Constantly open and allows things to go either in or out, and it's going to follow a concentration gradient. These are present to various degrees everywhere, okay? And then you've got a gated channel. Gated channels are gated. You need a key. So what is the key? Well, your key could be a chemical, or it could be a change in voltage, or it could be a change in mechanical shape. All right, you need to be familiar with all of these gates. These are the two that are most relevant for this chapter. Okay, the passive gates, the passive channels, passive channels, and the gated channels. All right, next, let's move to the next slide. All right, before we start looking at the pictures, let's talk about these two types of channel proteins. Okay, so type of channel proteins that go through the plasma membrane. We have these passive channels, which are also sometimes called passive gates, which is a little bit of a misnomer because technically they are always open, so they're an open gate rather than a closed gate with a key, okay? They are also called leak gates or leak channels, okay? The key component here is the concentration gradient. Things flow along their concentration gradient in and out of the cell. Okay? All right, and then you've got these gates with keys. Okay? And they're going to be a little bit different. We've already talked a lot about the chemical gate, which is also called a ligand gate, but people tend to forget what a ligand is. It's just a protein, okay? So these chemical gates are going to respond to a neurotransmitter, Okay? And then you've got the second gate that we've already talked about, which are voltage gates. Okay? And they respond to a change in voltage. So essentially, an action potential needs to be present. Actually, it doesn't have to be an action potential. I'm going to erase that. Any potential. It could be a local potential or a graded potential. I don't want you to get confused when we get to this component. So, basically depolarization. Depolarization needs to be high enough to reach a threshold that triggers the V gate to open. Okay? And then you've got one that we're not going to spend a whole lot of time on. These are mechanical gates. We'll talk about them a little bit when we do the special senses and when we do pain. Okay? And these respond to a change in shape of adjacent tissues. Basically, it's kind of like you've seen those little toys where you squish them and the little mouth pops open. It's kind of like that. All right, let's start looking at our pictures now. Okay, so I've got two major pictures here. What I like is right here, they are showing some passive gates or leak gates or leak channels. Okay, you'll notice that there is no door. These are open, and sodium is going to follow its concentration gradient, and potassium is going to follow its concentration gradient. Okay, then right here they're showing a voltage gate next to it. Okay, this one responds to a change in voltage. Okay, now when you are dealing with the plasma membrane, you will always have passive gates and voltage gates. If you're dealing with a motor end plate or dendrites, then you will also throw in chemical gates. And nearby, they will have passive gates. So it's not one or the other. These things are all sharing space and working together. Now in this next picture right here, I've got the voltage gate and then I've got a mechanical gate. Okay. And what you're going to notice right here is you see these little flipper arms right here and here. Those respond to the change in voltage, and that causes them to go like that, and then that causes this to open. Okay. But the mechanical gate, what you have are these little doohickeys here. And when you press, so say I'm going to press down right there. When I do that, that's like the little toy that causes this to pop down and go down. God, I hope that made sense. All right, I'm hoping that makes sense. So if you press on the tissue, it causes the little gate to pop open. All right, let's look at the next slide. You always, whoops, why did it do that? All right, what we're going to do here is look at our chemical gates a little bit more closely, and we've talked about these before. So we've used this picture where we see, we see a chemical gate here that is going to respond to a neurotransmitter. In this case, I'm using the example of acetylcholine, but there are other neurotransmitters that do it as well. All right, and they work by binding right here. When they bind, they alter the shape of the channel, causing it to open. And then when you use a enzyme to break them apart, like acetylcholinesterase, then it snaps shut and then things can't go through it again. Okay. Over here on this picture, the only reason I'm highlighting it is because we've got one that's sodium and one that's chlorides. And in both cases, they are reacting to the neurotransmitter GABA. Okay, but these are channels that let different things in. So this one lets sodium in, and this one lets chloride ions in. Okay, so that's kind of interesting. All right, let's look at the next slide. Now, in terms of voltage gate pictures, this is probably one of the more common ones that I'm using in this class. And basically what you're going to see is when the voltage changes here, that causes this to go up here, that causes that to go that way, and then sodium can go down the gate. Okay? All right, I think that's all I'm going to say right now about the voltage gates, so I'm going to end this recording.